In the previous modules, I have discussed about how a cell responds to different external or internal stimuli or the chemical cues. I also talked about different communication pathways used by prokaryotes, that is bacteria, or eukaryotes. I also talked about interkingdom signaling, and I also talked about the different exogenous and endogenous cues that are used by the barnacle larva, that is a dominant fouling larva, in their pelagobentic transition or in their metamorphosis. So, in this particular module, that is module 4, I will be talking about what are the different loopholes, why this is biocommunication in the oceans is handicapped and the reasons behind it, and what we should do in future. Basically, by using the advanced analytical tools such as flow cytometry, genomics and proteomics. These are the 3 areas which I am going to focus and discuss on. Now, deciphering the different biocommunication pathways was handicapped initially because of less than 1% of microbes can only be isolated using traditional culturing techniques. Now, 99% of the microbes cannot be cultured. So, it is very difficult to understand their biochemical pathways and they are inaccessible. Because in order to understand the different biochemical pathways, it is also important to know what are the types of microorganisms that are available, and that was not possible before. So, now in the late 1970s or 1980s, the Woods Hole Oceanography Institution scientists, they realized that there are some organisms which are less than 2 micrometer, and they are very important in the primary production in the oceanic or marine environment. So, in that case they thought and they tried to look for them in the epifluorescent microscopy or using different microscopy, but it was not possible for them. So, in 1985, Rob Olson for the first time he took a flow cytometer to the field, and this has created wonders in the field of marine ecology since then. Now, what is this flow cytometer consist of and what it is all about? The flow cytometer means cytometry means cyto is a cell and metry is measuring. So, it is a measuring a cell or cell properties when they are in a flow. You can see here it consists of fluidics where the cells flow, laser light which is targeted on these cells, electronics which detects all these different properties of a cell, and a computer which will give the scatter plot. Now, when a laser light strikes any cell, you can see here that it goes in this fashion, giving the size of the molecule. So, forward scatter actually gives the size of the molecule. If there is nucleus or there is any complex materials inside the cells, then it does not, this light does not go forward, but it is, you can see that it goes this way, that is it is scattered. So, this is giving rise to side scatter, that is the internal complexity. So, you can measure the different physical properties of a same cell when they are passing through a flow and interrogated by a laser light. So, now this way you can quantify the cells very fast, and you can also characterize them from the different heterogeneous populations into homogeneous subpopulations. And also there is one important characteristic of this is it also can sort. So, if I am interested in a particular population of a cell, then I can sort it in a tubes and I can work with those populations. Now, this flow cytometry offers also the ability to rapidly distinguish between active cells and dead cells. This is very important because what happens that in ocean environment only the active cells or highly metabolically active cells are involved in the processing of the organic matter, and bacteria plays a very important role in the organic matter production in the open ocean. So, it is very important to know not only how many bacteria are there, but how many of them are actually active and how many of them are actually dead population. So, this particular flow cytometer offers the ability to distinguish this type of cells, and it is very useful in the environment, understanding the effect of different environmental variables. Here you can see a classic scatter plot. This is a red fluorescence and this shows the green fluorescence. You can see the population is divided into P2 and P3. This green fluorescence are because of the live cells and red fluorescence is because of the dead cells. So, these live cells are stained by using Cyto 9. So, in the case of flow cytometry, the cells which are not able to fluoresce can also be tagged using certain fluorescent probes. So, in case of bacteria, you can see here we have used Cyto9 for probing the live cells and Propidium iodide to tag the dead cells. Coming to the next application of flow cytometry, that is flow cytometry with fish. Fish, everyone knows fluorescent in situ hybridization is used to identify specific bacteria. If I want to know a particular, for example, whether in a particular environmental sample Vibrio cholerae or E. coli is there or not, I can use fish. But this fish is very important tool in the molecular phylogenetic discrimination. So, this has been clubbed with flow cytometry because flow cytometry is rapid, specific, and it is also very fast, and you can analyze many samples together. You can also preserve the samples, and then you can analyze at a time. You can see that use this particular fish FCM technique to analyze different samples from different geographic regions in order to see what is the population of Vibrio cholerae in different geographic regions. So, in this case what is basically done is oligonucleotide probe, which is highly specific for a particular bacteria, is developed, and once it is developed using fish, then this particular bacteria are run on a flow cytometer, and then it will distinguish this bacteria which are very highly specifically tagged by a particular fluorescent probe, and you will get the results like this. Now, it is also important to know, this is another application of flow cytometry where it has been coupled with radioisotope tracers or radio labeling. It is also important, it was very difficult to understand who eats whom in a ecosystem models. Now, more than 25% of bacterial standing stock is grazed daily, and therefore, is a significant flux in the biogeochemical cycling of the carbon. So, these type of techniques, for example, flow cytometry and clubbed with the radioisotope labeling, it was possible to understand who eats whom. Also, it is also possible to understand the mixotropy in nootrophic microbes. These are only the few examples. There are so many other applications that are also possible by using this, but these are the few examples that I have cited, but this was not possible earlier, and also earlier it was not possible because of this lack of understanding the uncultured microbes. Now, coming to functional genomics and proteomics, which are gaining importance in the recent years, and this is basically dependent on the environmental DNA, wherein the DNA is extracted from the environment. So, you can unravel those microorganisms which could not be cultured. So, all the 90, as I said, only 0.1 to 0.01% of the microbes can only be cultured, and rest are uncultured. So, you can unravel the diversity of different microorganisms using metagenomics, and then it can be also transcriptomics and proteomics and metabolomics can be used for unraveling different biochemical pathways. Now, microorganisms, they inhabit or they exist in almost all the niches, and they are the ones which are actually responsible for producing different types of chemical compounds or chemical signals and bring about and help in different processes, for example, habitat selection, feeding behavior, etc. So, they are actually responsible for producing these chemical compounds which are the language of life in the sea. So, unraveling first their diversity and unraveling their proteome or metabolome will understand their metabolic capacities, will help to understand the different pathways which were actually handicapped before this. Now, combining the sorting capabilities, I said that a flow cytometer can also sort these populations whichever is of interest, and combining with the sequencing power of this metagenomics will actually improve the researcher's ability to target specific functional groups, and this is a step ahead in this field. As I said, metagenomics gives scientists access to millions of microbes which was actually not previously been studied. Now, there are peptides which are also very important signaling compounds in the aqueous environment, and these peptides or proteins can be sequenced or can be understood better using mass spectrometry. I have given, there are many other techniques which can also do that, but MALDI-TOF is one of the analytical tool which can be used to characterize the different proteins or peptides of interest. In this particular example, I have shown that this particular MALDI-TOF is used to identify the bacteria. Now, bacteria has got specific ribosomal proteins. This can be used as a signatures to identify a particular genus or a species of a bacteria. So, the bacterial proteins, ribosomal proteins can be extracted, can be processed, and then they can be sequenced, and the mass spectra thus obtained from there can be used to identify what type of bacterial species it is, because also as I said, it is very important to also understand what is the type of bacteria before understanding what is the communication pathway or what compounds or chemical they are secreting. Here you can see another plot where it shows MS spectra of different bacteria, but they are the different species. Genus is the same, but based on this mass spectra, it is actually possible to categorize them based on their species level. Now, deciphering these different biochemical pathways was handicapped basically because it was not, because it requires the augmentation of the experimental research, and this augmentation of the experimental research is possible only by framing some hypothesis and testing some hypothesis using the mesocosm experiments. Now, what are these mesocosm experiments? These are the experiments where the enclosures are used to enclose a particular environment, and this particular environment can be manipulated. For example, in terms of nutrients or in terms of introduction of some prey or prey-predator interactions can be studied. So many things can be studied. Energy transfer can be studied. Biodiversity can be studied. Adaptation. Most importantly, chemical cues and pathways can also be studied very effectively using mesocosm experiments. These are different examples you can see where these enclosures have been deployed. So, we have done one such experiment in the Eastern Arabian Sea. There is a mesocosm experiment in order to understand the importance of microbial loop. So, what was done was in the open ocean, the surface water was oligotrophic. It was having less of the nutrients. So, what was done was subsurface water which is rich in nutrients was translocated into the surface water. That means we manipulated that environment, and then it was seen how it affects the primary production, or how it affects the primary production, because now it is more eutrophicated. Now there are more of nutrients in this. This is one such experiment. That means deciphering different biochemical pathways. It requires augmentation of experimental research, as I said earlier, in oceanography, and these experiments can be used to test different hypotheses. This is one such hypothesis, and it can be tested also against microbe-metazoan interactions or unraveling different biochemical pathways and their role in the biogeochemical cycles and ecosystem functioning can be studied, and this is a step ahead in this field of biocommunication. With this, I come to the end of the module 4.